Hey guys, welcome back to Now I Know. Today we are talking about one of my favorite topic from immunology, and that is complement system or complement pathways. Now what we are going to do over here is we will understand first what are these complement proteins, from where does it come in our body, why are they called complement, and all three pathways of complement system, how they help us to get rid of an antigen or pathogen. Also, one more point that I want to tell you before I begin the video, I will give you some tricks here and there to remember the complement pathways in really easy, super fun, easy way. That is what I used to, I came up with when I was learning it for the first time in my postgrad. So let's begin today's video. So what are these complements? These complements are nothing but serum protein. That means they are the proteins that is present in our blood. Now, before I go on and tell you from where does it come in our body, let's see why are they called complement proteins. What scientists found, now this term is given by Paul Ehrlich. What he noticed is there are certain proteins in blood that actually completes the action of antibody. That means these protein complements the function of immune system. It helps the antibodies to get rid of an antigen. And here I'm saying specifically antibody because this is the initial stage of discovery where they were finding the complements helping antibodies. Now we also know that it also helps the immune system without antibodies. But this is what it was, the proteins that complements the function of immune system, and simply because of that they are termed complements. Pretty easy to understand. Now from where does it come in our body? With no big surprise, we know that liver cell hepatocytes play a major role in producing proteins that are secreted into the blood, right? So similarly, liver hepatocyte is the major source of complement protein. Now having said that, there are also few of the complement proteins are also produced by blood monocyte, tissue macrophages, and epithelial cells of gastrointestinal and genitourinary tract. So as I said, liver hepatocyte is the major source. If you know this much also is enough, but I think it's always better to know a little more about it. So a few of them are also produced by these sources. Alright, so now we know this much. They are the serum protein, why they are called complements, and from where they are produced. Now as I said, they are the serum proteins. That means they are always present in our blood. They are there in the circulation all the time. So if they have to be in circulation all the time, do you think it is feasible that they are always active, functionally active? It is not, right? Because if they are always functionally active, what will happen? First of all, it's waste of energy. If we don't have antigen and pathogen, we don't need them to be active. Also, they will attack our own cells, which is absolutely not feasible. So complements, they are present in the blood, but they are functionally inactive. Okay, now when we say that a substance is present in functionally inactive form, that means they are called proenzymes or zymogen. So they are present in blood in functionally inactive form, that is proenzyme. Unless they encounter or they have a trigger point, they will not get activated. And since they are in proenzyme state, when they have to get activated, also we know that they have to undergo proteolytic cleavage in order to be activated, right? So when they have a trigger point or when they encounter a pathogen, they would undergo a proteolytic cleavage to become active. Now all this is going to make sense once I show you the pathway. I just want to tell you quite a few points that we should have in background before we start with the pathway. Now again, one more point here. Once I say they undergo proteolytic cleavage, that means it is going to be broken down into pieces, right? And when these complements get broken down, it is always two fragments that they produce. Okay, let me put it down here. So let's say for an example, I'm talking about complement number 3, which is getting activated. That means it will be broken down into two fragments. One would be a smaller fragment and we would designate as A. That means it would be C3A, one fragment. and one would be a larger fragment. Let's designate this as B. So C3B. Now you can remember it this way, B for big. Alright, so B is the larger fragment and the other one is smaller, A fragment. Now generally what you will see constantly throughout all three pathways that we are going to learn about complement protein is that you will see that the fragment A diffuses away from the site of target pathogen. The B fragment would bind to the target cell. That means this larger fragment is going to carry out the destruction of a target cell, right? Whereas the A fragment is diffusing away from the target site. When we say that it diffuses away from the target site, that means it is not taking part at this particular target site, but it would take part in initiation of inflammatory response. So at this point I think you should remember only this much, that fragment A always diffuses away. Now you can remember it this way, A for away. So it is always the A fragment that diffuses away. A stands for away. B stands for big, that is a larger fragment, and you need the larger fragment to fight against the pathogen and it will remain at the target site. Now there is an exception in this. When I'm talking about complement splitting into two parts, there is an exception of complement number 2, and that means the exception is the smaller fragment is actually the B fragment and the larger fragment is the A fragment. So that means the larger fragment would stay. That means for C2, the complement number 2, C2A remains at the target site, whereas the C2B would diffuse away. It's an exception. Still some of the books do not show this exception. The books that I am referring and I have been studying from the beginning shows this as an exception, and that's how I have learnt it. So what you will see as C2B being the smaller fragment which diffuses away, and C2A is the larger fragment that stays at the target site. So this also, once we are done with the first pathway, I will tell you how to remember all these small points. Okay, so let's move on. Okay, so one more point that I think I should tell you is, as I said, they are in inactive form, right? Unless they have a trigger point, they'll not be active. So once they get a trigger point, it's not that all the complements are getting activated together. And so once one complement is getting activated, it is going to activate the next complement, and this activated complement will now activate next complement. So it will go on. That means they work in cascade fashion. So complement reactions work in cascade fashion. Once one complement is activated, it will activate the next one and so on. So yeah, I think these many points should be enough for us to understand. And now we can start with the complement pathways. Now there are three pathways. We will learn them all one by one. So let's begin with the first pathway. And for all three pathways, there will be an initiation point. This, I say, initiation point is different for all three. That means there has to be some reaction that is triggering the pathway, right? So for all three pathways, there will be three different trigger points. How they are activated, that one thing we should be clear. Then there will be a sequence, how these complements are coming, and the end product of all three pathways. Okay, and the end product is same for all three pathways, so it is going to be easy to understand. The trigger point is different for all three pathways. Then there's going to be sequence of the pathway that it follows, and the end product, or we also call this as MAC formation. We'll come to it. This is going to be the same for all three pathways. Okay, so let's begin with the first pathway. The first pathway is called classical pathway, and it is called classical because it is the first pathway that was discovered. Okay, now the trigger point for this pathway is, okay, let's start assuming that an antigen or a bacteria that has entered the body. What would happen? So let's take a bacteria that has entered our body. Somehow it has come inside the body. We know that there are certain surface antigens present on the bacteria that are recognized by our antibody, and it will bind to it. So let's take antibodies that are binding to this bacteria through this specific surface site. So now once this antibodies bind to this antigen, what would happen is, this is the Fc portion of antibody. We have talked about all this in one of the previous video when we were talking about antibody structure. So this Fc portion would now get activated and would expose the activated open site, which is now available for complements to bind. Alright, now what will happen? Now this results in binding of complement to this particular site. So first the C1 complement would come into the picture. Now the structure of C1, we have to understand the structure of C1 because complement 1 is made up of 3 subunits, that is C1Q, C1R and C1S. Again I'll tell you how you can remember all this. So let me put a diagram over here. As you guys know, I'm not that good in diagram, but still I will try. I know it's not the best, but it will give you some idea. So C1 is having 3 subunits. As you can see, C1 has this tail kind of a portion and this globular head over here. So the C1Q would bind to this Fc first through its globular head. Okay, C1Q would first bind to this Fc. Now, as I said, complement activation is in cascade. So what will happen is C1Q has now bound, that would activate the next subunit, that is C1R. So now the C1R has activated to act on C1S. So now when the C1R is getting activated, you can show it by putting a bar on it. That means now it has got the proteolytic activity to activate the next complement, right? So C1Q would activate C1R, which has now the serine protease activity that would activate C1S. So of course we can show it this way. C1S is now activated, so put a bar on the head. So let me put it in reaction form. We have antigen that has entered, antibody bound to it, that resulted in activation of C1Q. C1Q activated C1R, which got the serine protease activity, which activated C1S. Now the C1S has to activate the next complement, and the next complement over here is C4. Again I'll tell you how you can remember this order just once we are done with the pathway. Now the C1 has the enzymatic activity not only for C4 but also for C2. So it is going to act as the enzyme for C4 and C2 both. So what will happen? Let me put a target site over here. Now in approximation I'm putting, we have got C1Q, C1R, C1S activated. That is going to act on, the C1S is going to act on C4 and C2 both. Now let's come to our activation part. When I said that these complements, when they get activated, they get broken down into 2 fragments. That's what is going to happen over here. So now C4 would get activated. That means it will be broken down in C4A and C4B portion, right? As I said, C4A, the A fragment, A for away, it would diffuse away, but the B fragment would remain at the target site. So let's put it over here. After C1S is C4B, right? C4B comes and binds over here. So sorry, I'm not that good with diagrams, but I just want you to, have an idea what's exactly is happening. Now after C4 is done, it will act on C2. Same thing would happen, C2A and C2B. But do you remember what did I say before? C2 is an exception. That means B is a smaller fragment and A is a larger fragment. So here C2B is going to diffuse away, but C2A would remain at the target site. So let's put C2A over here, C2A. Alright, that is binding over here. This is our antigen. Okay, antibody is here. Sorry, I forgot to put over here. Here is the antibody, and to the Fc portion of this antibody, all this is tagged. Okay, so now what we can see over here is we have C4B and C2A, two fragments that are attached. Now this complex has gained the enzymatic activity for the next complement. Okay, this role of this C1 was activate its own subunit and activate C4 and C2. Okay, that's over. Now these two subunits together has gained the enzymatic activity for the next complement, right? So this again, to show the enzymatic activity, we put a bar over it. So we have C4B and C2A. Actually it is written as, C4B and 2A. You don't have to write C in between again. Now this complex act as C3 convertase. That means what? It has gained the ability to act on what complement? Simply C3 complement. So now this would split the C3 complement. We know what would happen again now. C3 would split in C3A and C3B. The A complement would go away, B would remain at the target site. So let's put this B over here. So what we gain here now? 3B is added. So now this is become C4B, 2A, 3B. Simple. Alright. Now this is again a complex to activate the next molecule, the next complement. So let's put a bar over here. And this is now C5 convertase. That means it can activate C5 complement by splitting it into two fragments, C5A and C5B. A would go away, B would remain at the target site. So let's add C5B over here, C5B. Right? So what is added over here now? We have C4B, 2A, 3B, 5B. The only difference over here, everything is B, only 2A is there because it's an exception. Again, I'll tell you how you can remember this. Now, till you have the C5 convertase formation and you have C5 complement splitting, it is different over here. After this, what I'm going to tell you now, the end pathway or the end product is going to be same for all three pathways. Okay, so once you have this formation, once we have this much from C5B, what I'm going to tell you is going to be the same thing for all three pathway, and that end part is called MAC formation. What is that? Just in a while we'll know. Now once C5B has come to this site, this becomes the attachment site for the next complement, and that is C6. So the next molecule, that is C6, would come and bind over here. Alright, and now it is very simple to understand. Once the C6 comes and binds, it will be followed by C7 and C8 molecule and poly C9 molecule. When I say poly C9 is the reason because there are multiple C9 which would come over here. More than one C9 complements would come and get itself, arrange itself in a cylindrical way in the membrane of the target cell. Alright, this C5B, C6, C7, C8 followed by C9, and to be very specific, poly C9, that means arrange itself in the membrane of a target cell in a cylindrical way, somewhat like this. See, now these are the C9 complements that have arranged itself on the membrane of the target cell, and this result in the pore formation on the membrane of the bacteria or pathogen. So that pore, because of the pore formation, there will be leakage of all the molecules from the cell, and that will result in osmotic instability. The cell will lyse, right? So because of this, this whole assembly is attacking the membrane of the target cell. It is called membrane attack complex, right? It is a complex that is attacking the membrane of a target cell. So it is membrane attack complex. And this happens once the C5B molecule is made, because this is an attachment site for C6, 7, 8 and poly C9. Alright, so this is called MAC formation or membrane attack complex. This is going to be same for all three pathways. Alright, now this pathway that we studied, we called it as classical pathway. As I told you, this is the first pathway to be discovered, and it involves the antibodies. Right? Now the antibodies that it involves are of class IgG and IgM. Okay, it's not randomly any antibody, but IgG and IgM. When it binds to the antigen, it is going to activate the classical pathway. Now, how will you remember this one that it involves only IgG and IgM is, look at this, we are talking about bacteria. This bacteria can be either gram positive or gram negative, anything. Okay, look at this, when we write it gram, this is how we write it, right? GM. That's where your clue is. It involves IgG and IgM antibody. Okay, so this is how you can remember. IgG and IgM, these two antibodies are involved in the classical pathway. And one more thing, if you look at the overall sequence of the complement here, you will find it, you know, let's just put it down over here. We have C1 followed by C4, C2, C3, C5, C6, C7, C8 and C9. What you can, if you look at this sequence carefully, you will see that only C4 is the complement that is little problematic over here. Otherwise the whole thing is in sequence actually. And the reason why this is not in sequence is, first of all, I will tell you, is because they are not labeled based on their sequence in the pathway. They are labeled based on their discovery. When they were discovered first, that's how they are labeled. So we didn't know function at that moment. Now if you look at it, as I said, only C4 is not in order. So now how I remembered this was Mr. C4 over here. Okay, Mr. C4 was too much in hurry to fight with an antigen. Okay, it just wanted to get activated first and go ahead and fight. So Mr. C4 jumped its queue and came ahead in the queue. But you know, you just can't go ahead of number one. Number one is always number one. So he had to sit after C4. Alright, so C4 came over here after number one because, as I said, he was like too much into, I want to fight with that pathogen, I want to get rid of this antigen and all that. So C4 sat behind number 1, and then it is the same thing, C2, C3, C4 has jumped of course, so C5, 6, 7, 8 and 9. Now what happened, you know, when this C4 jumped over here, Mr. C2 got really angry. He thought C4 is not following any rule. He just jumped from the queue, he jumped the sequence, so I am also not going to follow any rule. Which rule it didn't follow? It did not follow the rule of the fragment, smaller and larger fragments. So he said, I'm not going to follow this rule. My A fragment would be larger and B would be smaller. I'm not going to follow what others are following because C4 did not follow the rules. So that's why C2 is an exception, because he got really angry. Now, as I said, you might find some books which do not say that, you know, C2 is an exception. And it might follow just the similar rule, but the books that I refer, as I said, do show that C2 is an exception. That's why, you know, I just want to share it with you. Do not get confused if you see it the other way around. And here I said C1 has 3 subunits Q, R and S. How will you remember is, look at this, the pathway or the queue for complement starts with Q itself. That means C1Q. The queue of complements starts with Q itself. So C1Q followed by R and S. C4, because it was too much in hurry, so it came after C1, C4, C2, C3, 5, 6, 7, 8 and 9. Alright. Now look at this. Classical pathway, the trigger point is when the antibodies bind with the antigen. Because the antibody Fc site is the site where other complements are coming and binding. So it is antibody dependent pathway, much is cleared. Now if it is an antibody dependent pathway, this cannot be activated if the antigen is entering the body for the first time. Can this be a first line of defense? No. Because when the antigen or pathogen is coming for the first time in our body, we don't have antibody at that moment. It takes time for the antibody to develop. What happens when there is an antigen that is entering for the first time? This is not going to work because we don't have antibodies at that time. So there has to be an alternative to this particular problem, right? So that is nothing but the alternative pathway itself of complement system. So that's why it is called the alternative pathway. One thing that I like about this complement system is there are a lot of things over here, right? But the best thing that scientists have done is to name them in such an easier way that it's really simple to remember all this. So it's an alternative to complement pathway where it is not involving any antibody binding. That means it is antibody independent pathway. So now what happens in this alternative pathway? As I said, the trigger point for all three pathways are different. So there is not going to be any antibody, there is not going to be any C1 because C1 can recognize and bind to this Fc portion. You don't have antibody, you don't have C1 molecule. Alright, so what is the triggering point over here? How is this activated? Once again, we have to imagine any bacteria has entered our system. Now, as I said, bacteria would have certain surface molecules that are specifically recognized as antigen by our body. So what will happen is these molecules would be recognized as foreign particles in our body and that would trigger the response. Alright, so now what happens in alternative pathway? This is interesting. The C3 complements in our body has a tendency to undergo slow spontaneous hydrolysis. That means it can get split in C3A and C3B by its own. Okay, there is a spontaneous hydrolysis. It gets cleaved in C3A and C3B. Now we know that C3A is not a molecule or a fragment that is going to get attached to any target site. It is the B fragment. So C3B is a fragment that is in our circulation, right? Now, if there is an antigen, which is a new antigen that has come in our body, which is not going to get recognized by antibody as of now, the immediate reaction could be this C3b can come in touch with this foreign particle, recognize this as an antigen and would bind over here. So now the C3b has bound to this antigen because the C3b can recognize the foreign particle and it can bind to it. Now suppose there is no bacteria in the body, what is happening to this C3b? Because as I said before, we don't want any activated complement in our body, otherwise it is going to attack its own cell. So by chance, if there is no antigen and this binds to our own cell, this C3b fragment would get inactivated by inhibitory protein in our cell. It cannot carry out the further reaction. Okay, that's the beauty over here. But when it comes in contact with the foreign particle, there is no inhibitory protein. This antigen is not going to have inhibitory protein. That's the whole concept, that's the whole funda behind this complement system, right? So this C3b is now activated because now it has bound to an antigen. This would be now sensed by the next complement and it would come into the picture. So C3b, when it binds to foreign particle, foreign antigen, this becomes an attachment site for the next complement protein, and that is factor B over here. So factor B would come and attach to this site. This bound factor B can be cleaved by factor D. That means factor D can cleave this factor B. Again, we know once it gets cleaved, there will be two fragments that are produced, a smaller fragment that is BA and a larger fragment BB. Okay, so it is like this. Factor B is cleaved by factor D, and this is a bound factor B we are talking about. Bound to what? Bound to C3b, because C3b is activated. Why it is activated? Because it is bound to an antigen. Okay, so this is going to be cleaved in BA, that is a smaller fragment. Smaller fragment would diffuse away, and BB, so this BB is going to remain at the target site. So now this is what it is going to be. You already had C3b over here. This factor B is now cleaved into two fragments. A has moved away and there is only B that is remaining at the site. So BB. This is the complex at the target site now. Now this has to act as a complex for something else. So this is an activated complex. Let's put a bar over here. Now this is C3 convertase. Alright, this acts as C3 convertase. If it is C3 convertase, what it is going to cleave? C3 molecule, of course, into C3A and C3B. A would diffuse away, B remains at the target site. What would be the resultant complex over here? Let's put it down. I think I'm too messy in this video. So we have C3B, BB, which has cleaved C3. So we have again 3B. Again, it's an complex, active complex. So let's put a bar over here. Now it's going to be real easy. This acts as C5 convertase. Now this is easy to understand. C5 convertase acts on C5. You have C5A and C5B. C5A would go away. C5B is going to join the complex. So we'll have C3B, BB, 3B, 5B. Remaining story is the same as our classical pathway. As I said, once you have the C5 convertase formed, the rest of the thing, the end pathway is going to be same. Now C5B is going to be the attachment site for C6, C7, C8 and poly C9. That results in MAC formation. So the triggering portion was different and the initial series of complement was different than the classical pathway, but once you have C5 convertase made, it is going to be the same thing. So let's see once again what happened. As I said, this is the pathway where there is no antibody involved. So C3 has ability to undergo slow spontaneous hydrolysis in our body, so that results in two fragments, C3A and B. So what this C3B does is it tries and search a target site. If it is present, it can go and bind. So if there is an antigen that has entered the body, it is going to attach to it. If there is no antigen, it might attach to our own cell, but there is going to be inhibitory protein that is going to inactivate the C3B and thus it will not activate the pathway. But if at all it has attached to antigen, this is now going to be the attachment site for the next complement, that is factor B over here. Now this bound B factor is a substrate for factor D. So factor D can cleave this into BA and BB. BA goes away, BB binds to the site. So you have C3B and BB. This is a C3 convertase. That means it can convert C3 into active fragment. A would diffuse away. B is going to attach the target site. So you have C3B, BB, 3B. This is of course C5 convertase. C5 convertase can activate C5 into C5A, C5B. A diffuses away, B joins the target site. Now this is going to be attachment site for 6, 7, 8 and poly 9, results in MAC formation. Now how you can remember this, that alternative pathway has factor B and factor D in it. Okay, again this might sound silly, but this is how I used to remember and it helped me a lot. So now alternative pathway starts with an A, right? So A is taken up, right? So there is letter B that is available for a complement to take, so it becomes factor B. It could have started with factor A, but A is already taken in the name itself, so it starts with factor B. Next could have been factor C, but C has been taken up by complement. We are saying C1, C2, C3, so C is also not available. So the next best thing available was factor D. So you have C3 followed by CB because A has already been taken in the name. C is also been taken up in the name, so you have factor D. So that's all about alternate pathway or alternative pathway of complement system. Now the last pathway that was discovered, scientists found that it is getting activated without antibody and also without this C3B or factor B, and also the next pathway that they found is called lectin pathway. We'll come to it why it is called so, or it is also called as MBL pathway. So what was the trigger point here was, what scientists found was that it is a kind of a complement pathway that gets activated by presence of certain carbohydrate molecule on the antigen. As I said, the whole bacteria is not going to get recognized. There is specific surface molecule that are going to get recognized as a foreign particle. So what scientists found was that on antigen there are certain carbohydrate residue called mannose molecule, to be very specific, or menin molecule. This carbohydrate is recognized by a protein in our body as an antigen, and that protein is lectin. So lectins are actually this mannose binding protein that are present in our blood. So this was the pathway which was getting activated because this lactin which is present in our blood was recognizing this mannose on the antigen and was getting activated. And that is why it is called lactin pathway or MBL pathway, which stands for menin binding lectin pathway. Again, simple to understand, right? It is the lactin which is binding to the menin. Okay, so it is called MBL pathway, menin binding lectin pathway. So now there are certain pathogens that has this mannose on their surface. Okay, let me give you an example. Salmonella, Candida albicans, they have this mannose on their surface which is recognized by the lectin that is present in our blood, and this is going to trigger the pathway. So now what all complements are involved over here? Now when it comes to lectin pathway, it is very much similar to classical pathway when it comes to the initial portion. That means we saw in classical pathway we have C1 molecule that comes first, alright, and this C1 molecule has 3 subunits. So in lectin pathway, so instead of C1, Q, R and S, we have structurally similar molecule involved in this pathway. So this lectin pathway which has MBL, of course, so the first complement is MBL itself. That is nothing but menin binding lectin, because lectin is the one which is going to recognize and bind this. So scientists name that as simple as that MBL. So this is MBL that would recognize the mannose on the bacteria. This MBL is similar to our C1Q that we talked about in classical pathway. So it is structurally similar to C1Q that we talked about in classical pathway. So once MBL is bound over here, this is going to result in attachment site for 2 more molecules that are structurally similar to C1R and C1S. And those molecules are MASP1, MASP2. Don't be scared seeing the names, they are easy. This MASP stands for MBL associated serine protease. MBL we know is menin binding lectin. Now, so when you have MBL attached to antigen, that imagine as C1Q, you have C1R and C1S also coming into picture. So you have MASP1 and MASP2 becoming activated. Now from this it is going to be just similar as your classical pathway. Once this MASP1 and 2 is activated, this is going to be the activation site for C4 and C2 complement, just as similar to classical pathway. Once C2 is done, you will have C3 coming into picture, then C5, 6, 7, 8 and poly 9 resulting into MAC formation. The thing that is difference is only the trigger pathway, because there is no antibody over here. It is the mannose residue of antigen that is recognized by the lactin protein present in our body, that is MBL, menin binding lectin. Once this MBL binds to this antigen or mannose, to be very specific, this is going to activate the MSP1 and MSP2, that is nothing but serine protease associated with MBL. So MBL associated serine protease. 1 and 2 is bound, it is going to activate C4 molecule followed by C2, C3, just similar to our classical pathway. So when it comes to lectin pathway, all you need to remember is just the triggering point. After that, it is the similar thing as classical pathway. So that's all. I hope this was not too long a video, but I wanted you to have a comparison of all 3 pathways in one video, because that way it becomes really easy to compare and understand what is the difference between these pathways. And I hope these little tricks here and there helped you to remember and understand all the different terms and the sequence and all other points. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I'll see you next time. Until then, keep learning.